Welcome back to this next video in which we are discussing influenza. Okay, so we're currently in the process of discussing the influenza viruses, and the one that we've studied so far is the Influenza A virus species. Now, before we move on to the Influenza B virus and the Influenza C virus, I just want to again summarize some of these more complicated concepts that we saw at the end of the previous video. So, we saw in previous videos, we saw the structure of influenza A viruses. Now, I hope that's all very clear, but let's just go over this concept of serotype again. So remember, influenza A virus is a species of virus, just like humans is a species. And in that species, there are loads of different strains of influenza A virus. Now, all of the different strains of influenza A virus can be given a serotype, a class, of both hemagglutinin and neuraminidase, and overall we divide all of the hemagglutinin molecules of all of the different strains of influenza A viruses into 16 different serotypes, and the neuraminidase molecules, they're divided into 8 different serotypes. So for every strain of influenza A virus, you can give it a serotype of hemagglutinin and a serotype of neuraminidase, and this is what we mean, for instance, when we say that Spanish flu, which caused a pandemic in 1918, was of serotype H1N1. Now, the key thing that I want to stress, which is a major misunderstanding that people have, is that this does not uniquely specify the strain of the virus. There are loads of different strains of influenza A virus which all have this same serotype of H1N1, so it does not specify the strain. It is a case that the different strains of influenza A virus can be each given a serotype, but the serotype does not specify the strain. So there's loads of strains of influenza A virus that all have that same serotype. Now, hemagglutinin molecules and neuraminidase molecules that have the same serotype are similar to one another in structure, and they're similar enough to have loads of overlapping epitopes, and therefore, if you have two hemagglutinin molecules, even though they're different, when you get antibodies produced against those, if you look at all of the antibodies produced in response to one, and this is what this diagram is, and look at all the antibodies produced in response to another, there will be some overlap there. There will be some antibodies which can bind to both of those two different hemagglutinin molecules from the two different strains of influenza A virus. And if that overlap is big enough, then they are said to be of the same serotype. I do not know the actual technicalities of how you would take two influenza A viruses and decide if they are of the same serotype, or how indeed you would take a single influenza A virus and put it into a hemagglutinin serotype and a neuraminidase serotype. I don't know the technicalities of the experiment of how you decide which serotype it should be in, what sort of percentage overlap you need to have, things like that. I don't know the technicalities of that, but there will be technicalities like that. Now, one of the key points about influenza A virus is that it is capable of causing pandemics. Strains of influenza A virus are the strains of influenza that can cause pandemics. The other two species of influenza viruses, the influenza B viruses and the influenza C viruses, they don't cause pandemics. Now, let me just make sure everyone is aware of what this term pandemics means. I've been using it as though I'm assuming everyone knows what it means, but I'll just clarify that. A pandemic is where the disease spreads across a huge portion of the world. It's more than one country. It's spreading across a huge portion of the world. So a huge portion of the world gets the disease all at once, and it just keeps spreading and spreading and spreading. And the reason that you would get a pandemic of influenza is if there is no one in the population with immunity. If there are people in the population with immunity, then of course that hinders the spread of the virus, because if you've got a whole bunch of people who are immune to the virus, then they can't get it and therefore they can't spread it on. So that hinders the spread of the virus. So pandemics are caused when you have a virus to which no one is immune. Now, how does that occur? Well, it has to occur by new strains of influenza A virus that can infect humans suddenly being produced. So influenza A virus can cause pandemics because you can suddenly get brand new strains that have never infected any humans before unleashed on the human population. No one has immunity, and it just spreads and spreads and spreads across the entire world, and that is what we call a pandemic. Now, the reason that influenza A virus can cause pandemics and influenza B virus and influenza C virus can't is that there are so many strains of influenza A virus, whereas there are not so much variation in the strains of influenza B virus and the strains of influenza C virus. Both of those other species, influenza B virus and influenza C virus, they're going to have lots of strains as well, but there isn't as big a variation as there is in influenza A virus. So it's the massive variation of the strains in influenza A virus that means that you can suddenly create brand new viruses that can suddenly infect humans. Indeed, it's the fact that there is this huge population of influenza A viruses that usually can't infect humans, all of these ones here. They usually can't infect humans, and therefore people don't have immunity to these because they're not a problem. But if somehow you were to take one of these and make a modification to it that meant that it could suddenly infect humans, and we'll talk about this later on in much more detail when we come on to antigenic shift and antigenic drift. If you imagine it, you could take one of these strains that doesn't normally affect humans, make a modification to it to make it suddenly infect humans, then you'd have a brand new strain of influenza A virus that could suddenly infect humans and no one would have immunity to it, and that's when you would get a pandemic. And it's because there are so many influenza A virus strains, a huge number of which cannot usually infect humans, that you can suddenly get this brand new strain creation that can cause pandemics. So that's why influenza A virus can cause these pandemics. This is what happened in these 3 instances in the 20th century, and it was to horrible effect. Huge number of people got this illness, and a huge number of elderly people who then got this illness, it was bad enough to kill them. And that's what these 3 viruses are. Spanish flu in 1918, Asian flu in 1957, and Hong Kong flu in 1968. They were brand new strains of influenza A virus that could suddenly infect humans, to which no one had immunity, and they, spread through the human population and caused a huge number of deaths. Okay, the final complicated concept that I went over was this drawing here. So remember, there are a huge number of strains of influenza A virus. Not all of them can infect humans. A huge number of them can infect birds. Birds are really the major victim to influenza A virus. There are an absolutely huge number of strains of influenza A virus that can infect birds, but not humans. And then there are loads of other species that I haven't bothered to draw on this picture that can be affected by influenza A virus, such as non-human primates, dogs, horses, seals, ferrets, and rather usefully for experimental science, mice. But the one that I've put on here that is quite important and we'll come back to later is pigs. So when you look at all the strains of influenza A virus that can infect pigs, there is quite a significant overlap between those that can infect pigs and those that can infect humans, and also significantly there's a big overlap between those that can affect birds and those that can infect pigs. So keyly here, we have the possibility that pigs can become concurrently infected with a human strain of influenza A virus and some bird strain of influenza A virus that usually cannot infect humans. And then when you have concurrent infection by both of them, there is then the possibility that the genomes might get mixed up and you might be able to create a brand new strain of influenza A virus that might be able to infect humans, but which has a lot of properties of this avian influenza A virus that previously couldn't infect humans, and this is how we can create these brand new strains that can then cause pandemics. So pigs are believed to be the baddies. They're believed to be the place where new horrible pandemic strains of influenza A virus are created. Okay, so I just wanted to clarify that because I didn't feel that I'd explained these points as well as I hoped to in the previous video. So, we are now going to discuss the other 2 species of influenza virus, the less famous ones. Influenza A virus is the real baddie. It can cause really severe illness. It can cause these pandemics. It's the famous one. Influenza B virus is second most famous, and after that, influenza C virus is the least famous. But we're now going to have a look at their structures as we did for influenza A virus. We're going to see how they differ from influenza A virus. They do have almost identical structures, but there are differences. And we'll also talk, as we did for influenza A virus, about the different strains of the virus and the different animals that they can infect. Okay, so let's go over influenza B virus. So we'll do this down here, I think. So I'll just put a little title here. So we're now discussing the influenza B virus. So as I say, we will begin with the structure of the influenza B virus. What is different between the influenza A virus and the influenza B virus? Well, firstly, I should say all of the species of influenza A virus, they are all still going to be enveloped viruses that have a genome that is single-stranded, negative sense RNA and which is segmented. And in the case of influenza B virus, you're still going to have 8 segments of RNA, and they're still going to be packaged in the same way. You're still going to have a nucleoprotein, and you're still going to have the RNA-dependent RNA polymerase made out of these 3 proteins. So all of this is exactly the same. The M1 protein is exactly the same. The non-structural protein 2 is exactly the same. The M2 ion channel is the same. We have that again. Hemagglutinin is the same. Neuraminidase is the same. So what is different? I've told you nothing that is different so far. Here comes the different bit. In influenza B virus, you have another protein. In influenza A virus, we had 8 genome segments coding for 10 proteins. In influenza B virus, we have 8 genome segments coding for a whole 11 proteins. There is another protein in influenza B virus that we do not know the function of. It's another ion channel in the envelope of the virus, and it has the rather charming name MB. So, I don't know whether to actually draw this in on this picture. I will do it. We'll have some specific colour to show the modifications for influenza B virus. So here we go. In this ghastly sort of mustard yellow colour, here is this modification that we have to make to the virus particle to turn it into an influenza B virus. We have to put another ion channel in there, and I don't actually know whether it penetrates through the M1 shell here. I would imagine it does, even though I haven't drawn it there. But anyway, we don't know the function of it, so it doesn't really matter that I don't know whether it penetrates the M1 shell or not. However, in the envelope you have this new ion channel, and this ion channel is by the name of NB. Okay, so as I say, we don't know the function of this, but it is there. Now, the next thing to ask is, okay, we have this other protein, which genome segment is it going to be coded on? So, as I say, in influenza B viruses, we're still going to have the 8 segments of RNA, and you'll be relieved here that the naming system is exactly the same. So we still call these viral genome segments viral genome segment 1, viral genome segment 2, all the way up to viral genome segment 8. And absolutely lovely is the fact that the genes on each of the viral genome segments has been arranged, we've named them so that they are the same. So on viral genome segment 1 of influenza B virus, you still have the gene for PB1. On viral genome segment number 2, you still have the gene for PB1. PB1, etc. All of this is exactly the same for influenza B virus as it was for influenza A virus. The only thing we need to ask now is this MB protein, this to the ion channel here, which genome segment codes for the protein that makes this ion channel. And I don't actually know that much about this ion channel, so I don't know how many of the polypeptides have to oligomerize together to make the ion channel. It won't just be one protein, I wouldn't imagine, though. Okay, as you can probably tell, I don't know that much about this protein because its function isn't known, so at the moment it's not particularly important. However, I do know what viral genome segment has the gene that makes the polypeptide that is going to make this protein, and it is on viral genome segment number 6. So on viral genome segment number 6 of an influenza B virus, you will also have the gene for NB, as well as neuraminidase. You'll have NB there, which is the gene that will make this polypeptide that we can use to make this NB ion channel, which is in the viral envelope of the influenza B virus. So this is something to know about. However, it's not going to play any part in the pathogenesis story that I'm going to tell you, and that's not because it necessarily doesn't play a role, but we don't know at the moment whether it plays any role, so it's not going to play a role in my story, in my account of the pathogenesis at all. So it's just something for us to know about here. The other difference that I'm just going to point out about the influenza B virus is that the M2 ion channel of the influenza B virus is quite different to the M2 ion channel of the influenza A virus, and therefore it's important to call it a different name. So we call it BM2 to distinguish it from the influenza A virus M2 ion channel. So if you just put the M2 ion channel, people will assume you mean influenza A's M2 ion channel. If you want to specify influenza B's M2 ion channel, you should put BM2. As I said earlier, if you want to be very, very clear, you can put AM2 to mean the influenza A virus M2 ion channel. But if you just put M2, people assume you mean the influenza A virus M2 ion channel. Now, why is it important for us to distinguish between the influenza A virus M2 ion channel and the influenza B virus M2 ion channel? Well, they both do the same thing. They're both proton channels that are opened by a low pH on the outside of the virus. But the difference is that some of the antiviral drugs that we have, specifically a class of drugs known as the adamantanes, which we will come on to later, work by blocking the M2 ion channel. But they only block the influenza A virus M2 ion channel. They do not block the influenza B virus M2 ion channel or the influenza C virus M2 ion channel. So it is important to distinguish between the M2 ion channel of the influenza A virus compared to the influenza B and influenza C viruses, which is why we do indeed do that, because we have these drugs that are fantastic against influenza A virus. Well, I wouldn't say fantastic, but they are effective against influenza A virus, but they're only effective against influenza A virus because they block its M2 ion channel and not the M2 ion channel of influenza B or influenza C virus. But we'll come on to those drugs right at the end of the video. Okay, right. So those are the two structural differences that I wanted to point out about influenza B virus. Let's now talk about the different strains then of influenza B virus. So, just as with influenza A virus, there are a huge number of different strains of influenza B virus. It's a species and there are lots of different strains. However, thankfully, making it a lot more simple than the influenza A virus, we do not split all of the different strains into different serotypes of hemagglutinin and neuraminidase. There is only one serotype of hemagglutinin and neuraminidase for influenza B viruses. So what this is telling us is that if you look at every single influenza B virus on the planet, and you look at its hemagglutinin molecules and its neuraminidase molecules, they're reasonably similar in all the different strains of influenza B viruses. There is not as big a variation in the structure of the hemagglutinin and neuraminidase proteins in the influenza B virus population as there is in the influenza A virus population. And as I say, that is why influenza A virus can cause pandemics, because you can suddenly get brand new viral strains that can infect humans that are completely different from the normal strains that infect humans, have completely different hemagglutinin and neuraminidase molecules, and therefore the immune responses that we've had from previous influenza infections are no use at all against these new strains. Whereas with influenza B virus, it's less of a problem. It's not as much of a problem. So you don't get pandemics caused by influenza B virus. You don't get brand new strains that no one has any immunity to. Okay, so I'll just write a bit of this down then. So influenza B virus, it only has one serotype. So as I say, that tells us that all the different strains of influenza B virus have reasonably similar hemagglutinin molecules and reasonably similar neuraminidase molecules, so that there is at least some overlap in their antibody responses that they induce. Okay, but there are multiple different strains of influenza B virus still. The other important thing to say is that they do not cause pandemics. So I'll just write this down. Do not cause pandemics in the same way that the influenza A viruses do. Okay, the final thing that I want to tell you then about influenza B viruses is which species of animals can they actually infect, because they can't infect as many species as the influenza A viruses. So again, I'll draw one of these pictures to show the two major animals that can be infected that we know about for influenza B viruses. So here, this can be a set of all the different strains of influenza B viruses. So, of course, one of the hosts, which is why we care about it, is of course humans. So I'll draw this on here. So this bit can be the humans. So all of the influenza B virus strains that can infect humans are in this portion here, this subset of the whole set of influenza B virus strains. And then the other host that we know about for influenza B viruses is actually seals. So I'll put these on here. And of course there will be a little bit of overlap potentially between those that can infect humans and those that can infect seals. So here, this bit is the seals, like so. So this subset of the influenza B virus strains are the ones that can infect seals, and as I've tried to show there might be a little bit of overlap between strains that can infect humans and strains that can infect seals, so there will be some strains that are capable of infecting both humans and seals. Okay, and that's all that I have to say about influenza B virus. As I say, we've done most of the work when we talked about influenza A virus. All of these pictures that I've drawn for the in detail hemagglutinin and neuraminidase and M2 ion channel, they apply just as well for influenza B virus as they do for influenza A virus. So we really did most of the work when we was discussing influenza A virus, and thankfully there's much less variation in the population of influenza B virus, so we don't have to go into this complicated stuff about serotypes for influenza B virus. So that's actually important. I really want to nail that point home, that when you see people talking about, H1N1, they're instantly talking about influenza A virus, not influenza B virus or influenza C virus. That stuff is not used for influenza B virus and influenza C virus because there's not as much variation in the hemagglutinin and neuraminidase molecules in the influenza B virus and influenza C virus population. Okay, so moving on then to the next one. Let's do influenza C virus down here. Okay, so this one has, in my opinion, a more major difference in its structure compared to the influenza A virus, as compared to the difference between the influenza A virus and the influenza B virus. So, the first thing to say is that modification that we made for influenza B virus, that is not going to be there in influenza C virus. So forget this MB ion channel. That was a modification that influenza B virus had compared to influenza A virus. We're now starting afresh. Imagine that isn't there, and we've got our picture of influenza A virus. I'm now going to tell you about the modification that we make to influenza A virus to make the influenza C virus structure. Okay, so again, the genome is still going to be single-stranded negative sense RNA, but we're only going to have 7 segments in influenza C virus, not 8. So that's actually the first big modification. So I'll just put that here. So 8 genome segments. Completely wrong. 7 genome segments. That's the important modification from influenza A and influenza B. So 7 genome segments, and you'll see why we can have 1 less genome segment when we're talking about influenza C virus in just a moment. Okay, so let's go back to the virus structure then. So it's still single-stranded negative sense RNA, but we only have 7 RNPs now, and the ribonuclear proteins are still the same, so you still have the nuclear protein forming the capsid around the RNA segments, and you still have the RNA-dependent RNA polymerase made out of these 3 separate proteins, PA, PB1, and PB2. You still have the non-structural protein too, that's still going to be there. You still have the M1 proteins, you still have the M2 ion channel, although that's now called CM2, so I'll just put this down here. So we're going to have CM2 for influenza C virus, and it's going to be on a different genome segment as well, but I'll come on to that in a moment. The big change is now with regards to hemagglutinin and neuraminidase. Cross both of these out. They're going to be combined together in the case of influenza C virus. So influenza C virus has come up with a more efficient way of doing it than influenza A virus and influenza B virus. It says, I'm not going to bother having 2 separate proteins, one for binding and one for getting out of the cells. I'm going to combine them together into a single protein. So indeed, on the surface of the influenza C viruses, you have a single thing which resembles the hemagglutinin molecules, but which can do the activity of neuraminidase as well. So what we can imagine doing is get rid of the neuraminidase, and we will have this new thing that is going to be the combination of them on the influenza C virus that looks like this hemagglutinin molecule that we had on influenza A and influenza B virus. And this thing is going to be called the hemagglutinin esterase fusion protein, HEF for short. So I'm going to go over here then, and I'm going to draw this in more detail now. So firstly, let me write out its name in full. So as I say, this combination of hemagglutinin and neuraminidase that more resembles hemagglutinin to neuraminidase, I want to stress that it's not a mushroom shape like neuraminidase, it more resembles hemagglutinin to neuraminidase. It's just going to have the function of neuraminidase effectively as well. Although, as we'll see when we come on to the pathogenesis in more detail, it's slightly more complicated. It performs a different type of reaction to the neuraminidase, but we'll come on to that in more detail when we actually go on to the pathogenesis. You have to understand the replication cycle. You have to understand what reaction neuraminidase is actually going to catalyze before we can talk about how this reaction is going to be different. But at the moment, we can think of it having both the function of hemagglutinin and neuraminidase. So, HEF. This stands for hemagglutinin. Oh, and I should have said, again, I'm using a silly British English spelling here with the A in there. In American English, you'd get rid of the A, of course. Hemagglutinin, and then you put a dash. Esterase is the E, and that's, of course, an enzyme. And then it's the hemagglutinin esterase fusion, and you've got another dash between esterase and fusion. So this is the hemagglutinin esterase fusion, and then we might as well put protein after this. So let me draw out the structure then in detail of a hemagglutinin esterase fusion protein, and again it's going to be very similar to hemagglutinin. So there is a gene that codes for the protein that is going to make this protein complex, and again it's going to be a trimer, and this gene is of course called the HEF gene, and it's going to be on viral genome segment 4. In fact, I'll go over the modification to the genome before I draw out you the in-detailed structure of the hemagglutinin esterase fusion. So coming over to the genome here, we know that we're only going to have 7 pieces of RNA this time rather than 8. So luckily the first 4 are very similar. So number 1 is going to be exactly the same. We're going to have number 1 of the Influenza C virus genomic segment is going to be PB2. Number 2 is still going to be PB1. Number 3 is going to be PA. Number 4 is obviously going to change because it's not going to be hemagglutinin anymore. Instead, on number 4, you're now going to have the hemagglutinin esterase fusion. Okay, so in Influenza C, that's the modification. In genome segment 4, we've got hemagglutinin esterase fusion. Number 5 is exactly the same. We still need the nuclear protein, so that's fine. Number 6, of course, is going to be modified. We don't have the MB protein, because that's some silly rubbish that Influenza B virus has, and we no longer need neuraminidase, so this goes. This is no longer there. That's the one that we're getting rid of, number 6. And therefore, this one is no longer going to be called number 7. It's going to be called number 6. So I might put a cross through that one. This one is now going to not be called number 7 anymore, it's going to be called number 6, and of course it will contain the gene for M1, of course, and then CM2, which is the Influenza C virus M2 ion channel, which again is not sensitive to the adamantane drugs amantadine and rimantadine, unlike the Influenza A M2 ion channel. And then number 8, of course, is not going to be called number 8 anymore, it's going to be called number 7, and that still has the gene for the non-structural protein 1 and the non-structural protein 2 / nuclear export protein. So, Influenza C virus has 7 pieces of genomic material and only codes for 9 genes. So, to summarize, Influenza A virus had 8 pieces of genomic material and coded for 10 proteins. Influenza B virus had 8 pieces of genomic information and it coded for 11 genes, and Influenza C virus has 7 pieces of RNA and only codes for 9 proteins because of the fact that we have combined hemagglutinin and neuraminidase into one. So, on viral genome segment number 4 then, we have the gene for the hemagglutinin esterase fusion. So we synthesize this polypeptide, and again, just like hemagglutinin, the story is exactly the same as hemagglutinin. It is initially synthesized as one big polypeptide, which we call the hemagglutinin esterase fusion 0 polypeptide. What then happens is this is cleaved up into 2 separate pieces. So let me show this here. So it's going to be cleaved up into 2 separate pieces, one that's going to be globular, which is the hemagglutinin esterase fusion polypeptide 1, and then another one that's going to be less globular, and is going to be the transmembrane portion, which is the hemagglutinin esterase fusion 2. These are completely analogous to hemagglutinin 1 and hemagglutinin 2, so just drawing a picture of this, and I won't use the yellow colour because that was the colour we were using for Influenza B virus. So here is hemagglutinin esterase fusion 0, we cut it up and we get two polypeptides, hemagglutinin esterase 1 and hemagglutinin esterase 2, and they're going to bind to one another again by disulfide bonds, no longer by the peptide bonds, and then we're going to trimerize these together to make the full hemagglutinin esterase fusion protein that will be in the viral envelope. So let me now just draw this in here. So here is going to be the phospholipid bilayer, and now I'll do it in red again. Here is the trimer of hemagglutinin esterase fusion 2 polypeptides, like so, and I'll draw it in a nice cylindrical way here, like so, and then split this into 3 separate portions. Okay, so each one of those is a hemagglutinin esterase fusion 2 protein, and they are the transmembrane protein, so they're in the viral envelope. And then again, stuck on the top of each one of these hemagglutinin esterase fusion 2 polypeptides, we are then going to have the hemagglutinin esterase fusion 1 polypeptide. So I'll draw this here. One there, another one there, and another one there. So I've basically just drawn the exact same picture as I did for hemagglutinin. So these yellow circles are hemagglutinin esterase fusion 1. Now here comes the difference. Here comes the important difference. Each of the hemagglutinin esterase fusion 1 polypeptides will of course have a binding site, which I'll do here in purple, for whatever this molecule is that they bind on the surface of cells. And we'll come to that after we finish this bit on the viruses. We're going to come on to pathogenesis, and of course we'll discuss the viral replication cycle inside cells. So we will very soon find out about this molecule, don't worry. But at the moment, just know that those are the binding sites there in purple. Now, here is the difference. Each of the hemagglutinin esterase fusion 1 polypeptides, as well as having a binding site, is also now going to have an esterase site. It's going to have an enzymatic active site that is effectively equivalent to the enzymatic active sites of the neuraminidase proteins. And this is how it does both of the functions. So in green here, lower down than the binding site, so closer to the hemagglutinin esterase fusion 2, these are the esterase sites. These are the enzymatic active sites, so this is the esterase sites. Okay, and that is how hemagglutinin esterase fusion protein, the trimer, the whole thing, is different to the hemagglutinin trimer, that it also has 3 of these enzymatic active sites, so therefore it's going to perform the function of both hemagglutinin and neuraminidase. But don't worry, the life cycle, the replication cycle of influenza C viruses, thankfully, is still pretty much identical to influenza A and influenza B virus. It's just that we've combined both of the functions of hemagglutinin and neuraminidase into one nice protein complex here. Okay, so that's the structural difference then between influenza A viruses and influenza C viruses. We only have 7 genome segments now because hemagglutinin and neuraminidase have been combined into one protein that is coded for on genome segment number 4, so we've lost genome segment number 6, so genome segment number 7, which coded for M1 and M2, is now number 6, and NS1, NS2, which was genome segment number 8, is now genome segment number 7. Okay, those are the structural differences between influenza A and influenza C. Now let me talk a little bit about the different strains of influenza C, as I have done for influenza A and influenza B. Again, influenza C virus does not cause pandemics in the same way as influenza A strains can, and the reason is that there is not as big a variation in the population of influenza C viruses as there is in influenza A virus. There is only one serotype of hemagglutinin esterase fusion protein, and that does not mean that if you look at every single influenza C virus on the planet that they're all identical, that they all have identical hemagglutinin esterase fusion proteins. No, they don't. It's just that the variation is not that big that they don't have overlapping antibody responses. They all will have overlapping antibody responses, significant overlapping antibody responses, and therefore, again, we say that there is only one serotype of influenza C virus. So I'll just put this here. So again, for influenza C virus, we say that there is only one serotype, and the actual important thing to take from that piece of information is firstly that you don't talk about H and N numbers for influenza C virus strains, and also that it means that there is not as big a variation in the structure of this protein in the population of influenza C virus as there is in the hemagglutinin and neuraminidase proteins of the influenza A virus. There is massive variation in that in influenza A virus, and that's why influenza A virus is so dangerous and can create whole new, completely different strains that can suddenly infect humans with completely different hemagglutinin and neuraminidase molecules that no one then has any immunity to, and it sweeps through the population and causes a pandemic. But as I say, we'll talk more about the mechanisms of that when we come on to antigenic shift and antigenic drift, but it's a complicated concept, which is why I just want to nail it through as we go through the video. Okay, so one serotype of influenza C virus, and next piece of information, what different animal species can be infected then by influenza C viruses? What do we know about the hosts for influenza C viruses? So again, I'll draw one of these little set diagrams here. So if this box represents all the different strains for influenza C virus, so there'll be dots in here for all the different strains of influenza C viruses, as we have done for influenza A and influenza B already, then a subset will be those that are capable of infecting humans. So we'll have this over here. So here are all the influenza C virus strains that can infect humans. Then, what other animals are there that can be infected by influenza C viruses? Well, the other big host for influenza C viruses is pigs. So I'll draw this on here, and again, there'll be a bit of overlap here. So this, on this side, this is the subset that can infect pigs. And as you can see, there's a bit of overlap between the subset that can infect pigs and the subset that can infect humans. So there'll be some influenza C viruses that can infect both pigs and humans. So there are fewer species that can be infected by influenza B viruses and influenza C viruses is the key message here. Influenza A virus is the really, really difficult to understand one and the really, really dangerous one because there are so many influenza A viruses and they infect so many different species, birds being the prominent victim of influenza A viruses, whereas influenza B viruses and influenza C viruses, there's much less variation in the population, and they can infect, we know that they can infect far fewer species, there are far fewer different strains, and the pools of species that they can infect is much smaller, as I have shown with these diagrams. Okay, so that concludes this portion of the video on the actual influenza viruses. I hope you now understand that there are 3 different species of influenza virus, that they do have different structures, that in each species there are a huge number of different strains, particularly for influenza A virus, and the variety of different strains of influenza A virus is incredible. They have hugely different hemagglutinin and neuraminidase molecules, and this is how we categorize the different strains of influenza A virus into serotypes, where all the influenza A virus strains in a single serotype have similar hemagglutinin molecules, if we're talking about a hemagglutinin serotype, and similar neuraminidase molecule, if we're talking about a neuraminidase serotype. Whereas we don't do that for influenza B virus and influenza C virus because all of the different strains of those 2 virus types have similar enough hemagglutinin and neuraminidase molecules if we're talking about if we're talking about influenza B virus, and similar enough hemagglutinin esterase fusion proteins if we're talking about influenza C virus, that they will have overlapping antibody responses. So we therefore think that there's only one serotype of each of those species. Okay. We will call it there for this video. In the next video, we will begin our discussion of the pathogenesis of influenza, and we'll start the discussion of the viral replication cycle, how it enters host cells, how it uses those host cells to make more influenza virus particles. So I'll see you in the next video.